Welcome back. Hello everyone. In our last lecture, we discussed about how to download FASTA file of a gene from NCBI. We downloaded this file. So today we will teach you about how to read, how to parse this file through Biopython. So I am creating a new Python file SeqIO .ipynb file. Okay, so first of all I will go to install biopython. Pip install biopython. Okay, after running this file, okay, it's already satisfied my library, means it's already installed in my system. So for this I will demonstrate two different way. First one, first method. Okay, my first method is from bio import SeqIO. Okay, so after that I will read this file by name SeqFasta. SeqIO dot read by read method sequence Fasta, and I will notify the file format in Fasta format. Okay, after that I will go for print SeqFasta.id. It will give me the NCBI accession number of this gene, and also I will go print SeqFasta.sequence. Okay, so for this I will go for str. Why str? Because sequence nucleotide is string format, so I will go for it. So this is NCBI accession number with chromosomal start point and chromosomal ending point. So below is the sequence. My second method, same from bio import SeqIO. Okay, for this I will go for again seq fosta equal to SeqIO dot parse. After that I will give my file path and then first a format of the file. After that I will go for loop for seq and seq faster. I will go for identifier. We can say id equal to seq dot id. For sequence we can say is sequence equal to seq dot seq. So let's see we can start seq dot seq or not. Let's check it. So run it. Okay, it's running. So after that I will run id Okay, and then I will print my seq. Okay, so this is sequence accession number with start and ending position, and this is the sequence. So this is the two different method. You can analyze by many other method and SeqIO. So this is enough for today. We will demonstrate each and every thing soon. Happy learning. Bye. Take care.